Hi, everyone. I hope all of you are well and healthy. My name is Warren Alling from the Spinal Cord and Brain Injury Research Center at the University of Kentucky. Today, we have really two great sets of talks that I think will be helpful to all of us. Thank you for joining us. But before I introduce Michael Orr, our moderator for today, I'd just like to take a quick moment to acknowledge all the graduates of 2020. These are the graduate students, the undergrads, clinical students, the medical students, PT students. I say this now because here at UK, tomorrow was to be our commencement day. And so while we might not be together physically, we're all together in spirit. And so I just want to acknowledge all of those folks who are graduating this spring. Great job. All right, so moderating today is Mr. Michael Orr. Michael Orr is from John Genzel's lab here at UK, and he is going to help introduce today's speakers. Michael, it's all yours.
Great, thanks, Warren, and thanks for putting all this together, everybody that's been working on it. I'm pleased today to introduce Dr. Abel and Abel Torres Espin and Dr. Austin Chou, our first speakers, followed by Dylan McCreedy a little bit later on. And the first talk today is going to be from two postdoctoral researchers at Adam Ferguson's lab. They work in the Brain and Spinal Cord Injury Center, or BASIC, at the University of California, San Francisco, and at the Zuckerberg San Francisco General Hospital. So first, we'll hear from Dr. Abel Torres Espin. Abel obtained a PhD in neuro cell therapy for spinal cord injury models from the University Autonomous of Barcelona under the supervision of Xavier Navarro. He then moved to Kareem Fouad's lab at the University of Alberta, where he studied the crosstalk between inflammation and rehabilitation after spinal cord injury in animal models. Now, Abel is exploring the intersection between computational and statistical methods and neurotrauma, and he focuses on data-driven discovery and data sharing in neurotrauma. After Abel, we'll hear from Dr. Austin Chou. Austin completed his PhD in neuroscience with Susanna Rossi at UCSF, where he studied pathophysiology of traumatic brain injury with specific interest in immunology and aging. Before joining the Ferguson lab, he interned in industry as a clinical data scientist working on neurodegenerative diseases. Now Austin is focusing on bridging preclinical and clinical molecular and computational approaches to accelerate translational research. Abel and Austin will be talking to us today about the Open Data Commons for spinal cord injury, a new data management platform that they have helped design and develop as a digital tool for the spinal cord injury research community. And with that, Abel.
Thank you, Michael. I'm really thankful to be here. I think I speak for Austin as well, and pleased to see that the community is coming together in these sort of like trying times around this online seminar. So thank you to everyone that is involved and everyone that is listening. So as Michael was saying, we're going to be talking about a community effort around data sharing in the SCI research, and specifically about the Open Data Commons for spinal cord injuries, this website platform that we have been developing in union with the community to facilitate data sharing in spinal cord injury. So I'm going to give a brief introduction of what the Open Data Commons is, and then Austin will give the updates and sort of a demonstration of all the new functionalities that we've been incorporating in the Open Data Commons. So we are all familiar with the data journey when we are in the lab and collecting data and generating data. And at some point, as we progress on that process, we condense that data, hopefully to be able to publish in a few pages in a narrative form. And that's only if we are lucky enough, because we all know that there are publication biases that sometimes prevent us to publish our research. All of this sort of like model of data dissemination, of knowledge dissemination, produce several issues, and that I don't have time to go in detail, but the main problems is that it's been shown that it's a waste. You can easily see that affects reproducibility and prevents data discovery. So one of the solutions that it's been there for a while, but not as much in the biomedical sort of like research, is data sharing. And we believe in the sort of like the spinal cord injury community that there are two things that are essential to reach the goal of, or break the challenges of data sharing in our community. On one side are the social things that needs to change, the cultural environment, so like bringing the community together around the idea of data sharing. In the other side are the tools and the sort of like the technical things that allows us. And one of those tools is the Open Data Commons for Spinal. So the goal or the mission of the ODC-SCI is to promote exchange of data, tools, and ideas in order to sort of accelerate the treatment and cures for spinal cord injury. But we do that using a specific framework. It's the first share principles. So first share data, or the FAIR guiding principles for scientific data management and stewardship, is that got published in 2016, is a very highly cited article that it's been picked up by journals and funding agencies, sort of like the requirement, the principles that we need to follow when we release data, that it's something that is being required more and more often. So FAIR means that data needs to be findable, meaning that humans and machines should be able to locate that data. Data should be accessible, meaning that once you locate the data, we all should be able to access that data. And also FAIR means the data needs to be interoperable and reusable, meaning that it contains enough structure and enough information so that we can operate that data with different tools, or we can, for instance, mingle that data with other data sets, but also it contains enough information that we can use so like to understand and describe the origin of the data. So to bring the concept of FAIR to the open data, to the SCI community, we there was a meeting in 2016 with sort of like the goal of introducing FAIR to the SCI researchers and other members of the community, called Developing FAIR Share Community. And so sort of like from that, we presented the challenges of FAIR and data sharing for spinal cord injury. Later on, there was a second meeting called FAIR SCI Head, where we sort of like got deeper on the details and assessed the readiness and so like the next steps. From that meeting, we got a roadmap to so like answer the needs of the community and to adapt FAIR to spinal cord injury. After that, a year after, we hosted the Street Fair. So like the Street Fair meeting, where we gathered the community and got feedback from users to develop the ODC-SCI. So during all these years, this sort of like gathering information from the community, it's been implemented in the ODC-SCI. So currently the site or the project, the initiative, it's managed from like these 4 teams that we have here, the leadership team and the data team that sort of like do the development and the daily operational part of the ODC-SCI. We have an executive board that oversees our activities, and a community board that we're still working on filling, and that the role is to sort of like keep up with the community around what's going on with the ODC. And with that, I'm gonna let Austin to go ahead.
Great, thanks for the introduction, Abel, and thank you, Michael and Warren, for the opportunity. Hope everyone can hear me. So the current implementation of the ODC-SCI has several different purposes. Of course, we hope to be able to help streamline the dataset publication process for all of you, as a lot of funding agencies and journals are now requiring you to publish your dataset when you publish your articles as well. As Abel mentioned, we want to promote those FAIR principles of data sharing. And importantly, we really want to promote open data and open science. And towards that end, we want to offer the opportunities to share unpublished data and published data in a protected way where you can get credited for your work while really contributing to the greater scientific community. And of course, we also want to offer the means to be able to use the ODC-SCI as a data repository or as a way of organizing the data you've generated in your own lab for long-term storage. But most importantly of all, as Abel pointed out, we've developed the ODC-SCI with constant feedback from the SCI research community, and we continue to hope that you'll work with us as we mature this site together. A lot of you might be familiar with the older implementation of the site, and so I just want to kind of highlight some of the update goals we have with this current implementation. First, we wanted to make it clear how the ODC-SCI is organized and what you can expect to see on the site. We also wanted to improve the interface to make the entire process much more intuitive to navigate from registration all the way to the data workflow that I'll touch upon shortly. We also added new functionalities that will promote these FAIR principles. Things like the ability to upload dataset associated documents like your methodology, data dictionaries, and also to be able to assign metadata to each specific dataset. That metadata will make it easier for people to interpret and understand where that data came from. For example, listing authors, contributors, funders, and writing an abstract, much as you would write an abstract for a paper. So in brief, there are several different account types on the ODC-SCI website. These different account types offer different accesses to the site or different available functions. You can see that someone without an account can see, explore, publish data sets, but they actually can't even download those yet. To become a registered user, the next step in the account types, you have to register with us, which just requires a valid email, in which case you can then explore and also access or download published data sets. Of course, we encourage everyone to then go on to join the ODC community. To do so, you'll actually be given a button to hit as a registered user saying, join the community. This will actually send a notification to the ODC committee asking them to approve your request. And once you're approved, you'll have the options to join the lab. If you're a PI and you want to register your lab, that will require another round of ODC committee approval. Or if you're trying to join an existing lab that already has a space on the website, that'll actually send a notification to the lab's PI so they can approve you onto their lab. And you can see that when you're a lab member, that's when you truly have full functionality of the site to be able to upload, share, release, publish your data, and also be able to explore and access data that is potentially unpublished but shared to this SCI community. So I briefly mentioned the idea of a data workflow. The ODC is organized into different data spaces, and these data spaces dictate who can actually see and access that data. In short, when data is first uploaded, it's uploaded into this personal data space. Then the PIs can trigger the process of sharing this data to a lab space, releasing that data to the commons, and publishing that data, generating a DOI in the process, which I'll touch upon more later, and releasing that into the public, or publishing that into the public rather. You can also see that each of these different data spaces also correspond to those different purposes I mentioned before. You could use the lab space as a means of organizing the data sets in your lab. Of course, data sets that are published into the public space will go on to meet those funding and journal requirements. And we really hope to build out this commons data space as a means of sharing data among the community in this protected way towards the goals of open data and open science. And just to give you a brief summary about how data spaces correlate with data accessibility, you can see that when data is first uploaded to the personal space, only the original uploader and the lab PI can see that data. When the PI shares that data to a lab space, then other members of that lab specifically will also be able to see that data. Data that is released to the commons is then visible and accessible to those who are ODC general members. And lastly, the data that is published into the public will be accessible by registered users and visible to those even without an account. So I would love to take a moment for questions. While I'm fielding some questions, like a few minutes, I encourage everyone to visit the ODC-SCI website and begin the registration process while you're listening to the rest of the presentation.
All right, so it looks like we have a few questions. The first one's from Caitlin Hill, and she asks, is the data restricted to in vivo work, or can in vitro work data be uploaded as well?
So we have no restrictions on the type of data, and we are happy to work with you to format your data or really work with your data for data upload. So yes, in short, we'd be happy to take in vitro data as well.
Great. It looks like we have another one coming in from Jill Ward, and she asks, annotating contributors in the site could be a way to help trainees get credit for some of their work, even if they haven't received authorship. Is there a way for them to do that on the site?
Yes, so that's a great question. We understand that crediting people for their work is extremely important, especially as so many of us want to have this as a component in our scientific careers. And so in the process of even releasing data to the commons, you're able to generate these DOIs, or digital object identifiers. And in the process, that generates citations. So when other folks want to use your data, they'll cite your data set as a work product. And so definitely trainees, and just anyone really in general, will be part of that citation.
Okay, one more just came in from Cassandra Alexandropoulos. I'm sorry, Cassandra. Would technically challenging protocols for spinal cord injury research be able to be uploaded as well, along with comments of what worked and what didn't work?
So I briefly touched on, but I won't go into too much detail in this presentation, on the idea of being able to upload dataset associated files. For example, that methodology document is actually quite free form. You can upload quite thorough protocols along with notes that you have about what works or didn't work. As well, in that in the metadata I mentioned before, you can write in the abstract, for example, notes about specific conclusions, including what didn't work, as that I think many of us recognize that publishing, or at least sharing with the community what didn't work, is just as important as sharing what did work.
Okay, it looks like we have some answers coming
Yeah,
in.
so I think
on
you
the
continue
chat.
on. I think Michael's frozen in time.
Oh, okay. So I'll move on. Happy to take more questions. And we have a lot of our executive committee, data team, and community board members in the chat right now. So I'm sure they'll be happy to field as many questions as you have. So for the purposes of the demo, I just want to quickly go over the registration process again, really, really fast, just so you know what to expect as you're stepping through these different account types. I want to point out where you can find help on the site, but I want to spend most of the time going over that data workflow, from going from upload all the way to publication. And along the way, we'll touch on the lab management for PIs. Hopefully, some of you have visited the site, and you'll see that this is the front page. On the bottom left are links that go to these organizational summaries. You'll see a lot of those figures I already presented there as well, just to remind you about how we've structured the ODC-SCI. When you hit create an account, starts the registration process. When you're a registered user and log in, you'll be given the Join ODC Community button. When you're approved by the committee, you'll be a general member and have the opportunity to join or register a lab. Once you're a lab member, you have access to the entirety of the site. This is your view, or what is known as your dashboard. Here you can see all the labs that you belong to, as well as all the data sets you've uploaded, including which labs they were uploaded to, as well as what their status is in that data workflow. For help, on the left-hand navigation bar, you can find the general help FAQ and tutorials links. The tutorials include step-by-step guidance or step-by-step instructions about how to format your data or how to upload the data, and so on and so forth. You're always welcome to contact us using the Contact Help Desk button in the bottom right of every single window. That we highly encourage everyone to help report bugs, or if you have any suggestions just in general, please feel free to contact us using the help desk. So let's get started with the ODC workflow. To start the upload process, on the left-hand navigation bar, you'll see the Upload New Data Set link. This will take you through the upload data process, which really has 4 major steps. You want to make sure you're uploading to the right lab. You choose your data file, and again, there's documentation about how to structure your data and format it for upload. I want to briefly touch on this idea of common data elements. It is a system we are still currently implementing, so when you're uploading your data, please feel free to just move along this step. But in brief, common data elements, or CDEs, are important and very helpful for making data fair, as common data elements provide standardized terminology for the field of research. Hopefully, we'll work a lot with many of you to formalize this system, and we'll also be happy to help work with you to map your data to these common data elements. But once your data is fully uploaded, you'll be able to see the data on the ODC site. This is just a quick view. I won't spend too much time on the dataset page, but you can see that the status is in the top right. For example, this newly uploaded dataset is currently still in the personal space. There's a few buttons for those data-associated documents and metadata, but please feel free to visit the help and tutorial pages for more information. So how do we actually go about sharing, releasing, and publishing a dataset? I mentioned that this is a function that is given to the PIs. And so PIs will actually be able to get to this Manage Lab page using the left-hand navigation menu again, finding Manage Lab there. When you're ready to share, release, publish your data, you simply find the dataset of interest on the bottom window. You click that dataset status, which opens up a dataset status window. Here you can see different statuses that correspond to those data spaces I mentioned. Sharing to lab members, shares into the lab space. Release to ODC-SCI community, releases to the common space. And lastly, when you request a DOI, that initiates a publication process to move a dataset from the ODC towards a public space. In brief, DOIs are digital object identifiers. They're accessioned through the California Digital Library, and they're uniquely assigned to every single dataset that is published. As I mentioned, when you want to publish a dataset, you open that dataset status window, and you basically select Request DOI, Publish, and Submit. For now, we also highly encourage you to email the team at info@odcsci.org to just make sure that we get your request and that we can address your request in a timely manner. More importantly, when you're requesting a DOI and publishing a dataset, it initiates a quality control process. Our data team will work with you to make sure that your metadata and your associated documents are of quality that upholds those fair principles of data sharing. And again, we really, really want to work with you on these items. And so please feel free to ask questions. Please feel free to contact us. And in the future, we'll continue to build out the DOI request system to be even more integrated with the current interface. And just to show everyone what an example of a published dataset looks like, this is one of those published datasets on the ODC-SCI. This is the metadata and citation page. You can see that, as I mentioned before, there is a citation that is associated with this dataset. So when other third parties or other labs go on to use this dataset, they'll cite this dataset much as they would cite a paper, right? So generating a work product that you can point to throughout your career. This published dataset also has an excellent abstract. It's quite detailed in terms of what the original purpose of the study was, how the data was collected, and even a primary conclusion that the researchers originally drew from this data. And as mentioned, here is a lot of the metadata, contact information, which lab generated this data, funding acknowledgements, contributors, which can include, for example, trainees. And so really this is the end goal of that workflow. And just to give folks an idea of the numbers that we have currently, we have over 50 registered labs on the ODC-SCI. We have over 200 registered users, and there are currently over 200 uploaded data sets. We understand that a lot of the data sets right now are primarily still held in that personal space, which is very limited to just the original uploader and the PI whose lab that data belongs to. We hope that with this new iteration of the ODC-SCI, the process of moving data towards a lab space, towards the common space, and towards publication is even more intuitive and we'll hopefully see these numbers in the common space and the public space increase. And just to give you an idea of the international community here, we have visitors from over 45 countries, and these are recurrent visitors. People are coming back. And we hope that we'll continue to build this community with all of you as well. So for now, we aim to continue to collect feedback. We're always interested in more feedback, and we would love to have any suggestions you have or any bug reports you might stumble into. You can contact us using the help desk, as I mentioned, or email us directly. We're currently working on further formalizing and integrating the DOI request and publication process, but those processes are live. And we're always looking for more opportunities to do more outreach and education about data sharing, especially for the community. And so in brief, these are the acknowledgements. We are currently funded by the Craig H. Nielsen Foundation and Wings for Life. We have a grant with them for the next few years. We're also supported by the VA, NIH, the Rick Hansen Institute, and the ISRT. And really, this is a call to action. If you are interested in participating, please go sign up. And we'd be happy to discuss more and work with you closely towards the future of the ODC-SCI. And I'd be happy to take any questions. Thanks everyone for your time.
Great, thank you very much, Abel and Austin both. So it does look like we have a few questions coming through. A lot have been answered on the chat if you want to go back through and look. But one that came up is Claire Francesca Mihan asks, it's similar to Caitlin's question, but is this restricted to spinal cord injury, or would spinal cord physiology in general be welcome?
Abel, do you have strong feelings? I think we are happy to take. We're happy to help upload and publish data sets generally. So
yeah, so the original idea of the ODC-SCI moves around spinal cord injury, and I will say any data that it's related to that. So spinal cord physiology, it's definitely related with, you know, the normal function of the spinal cord. It's related to the injury and the damage, right? So we will be happy. I think I speak for myself here, but I think there is no sort of like crawling or hosting that kind of data.
Okay, great. Another question comes in from Deborah Hemmerly. Is there a way to share qualitative data such as free responses that aren't categorical?
So there might be some more work needed to format the data appropriately, but the data structure that we use can certainly take qualitative data. It's more a question of how do we structure that appropriately. And again, if you reach out to us, we'd be happy to work with you on that.
Yeah, just as a note there, there's been a few questions about formatting data and what kind of data it's possible. If you can think that your data can be formatted in a spreadsheet, you know, like Excel or something like that, that's something that we can take in.
Okay, great. And one other from Lawrence Moon. He asks, the website has some good videos to explain how to upload the data. Are there any videos about how to interrogate the data?
I believe not. We don't have that yet. But that's a good point. We're working on having tools for being able to interrogate the data, and we're looking for having those tools and then building material and tutorials for that.
Okay, and I think we have time for one last one. So from Noam Harrell, is there an effort to accumulate the disparate data sets and metadata into a more comprehensive or universal database?
I see Adam respond to that question. And the answer is yes. I think that's right on point.
Fantastic. So again, I'd like to thank Abel and Austin both for giving us a great talk about a new tool that we can use for our data. But now we have another talk coming from Dr. Dylan McCreedy about another tool. He's going to be talking to us about tissue clearing and 3D light sheet imaging of the intact and injured spinal cord. So Dylan, oh, I lost my place here. There we go. Dylan earned his PhD in biomedical engineering from Washington University at St. Louis with Dr. Shelley Sakayama Elbert before starting his postdoc with Dr. Lonnie Shea at Northwestern University. Next, he moved to Dr. Linda Noble and Todd McDevitt's lab at the University of California, San Francisco and Gladstone Institutes, where he used 3D imaging technologies to study acute inflammation after spinal cord injury. Dylan first landed in Texas with Dr. Cliff Lowell studying neutrophils in acute spinal cord injury. And now Dylan is an assistant professor at Texas A&M and is a TIRR Foundation faculty fellow. With no further ado, Dr. Dylan McCreedy.
Hey, Michael, thank you for the introduction. I really appreciate that. And thank Warren and the rest of the steering committee for the opportunity to present. So I'm going to present some work that I performed mostly at the latter end of my postdoc and has continued on and become a major part of my lab. And I've updated the title a little bit to emphasize the passive tissue clearing techniques that we're exploring in my lab. So in order to keep the tradition going, I wanted to first show the pets that we have with us right now, our cat Tater Tot and our dog Gunny. They're helping us keep sane while we're stuck at home working all the time. And I'd like to thank up front all the people that have been involved in this project. This work was done primarily in Todd McDevitt's lab while I was part of Todd's lab, as well as Linda Noble's lab. And then thank the members of Todd's lab that contributed to this. I'd also like to thank the members of my lab that have participated in this, in particular, 3 outstanding undergrads that we've had, Megan, Hannah, and Maddie. They helped me get this project up and running this summer after I started in Texas. I'd also like to thank the imaging core here at Texas A&M, the MIC, which have gone a long ways to helping us get light sheet capabilities at Texas A&M that my lab uses all the time now. And then I'd also like to thank all of my collaborators. Today I'll be showing work from specific collaborations with Stephen Crone, Jen Doolin, and the late Wes Thompson. And then thank all the funding sources that have been involved in this project over the many years. In particular, thank the TIR Foundation, which created the funds that made possible several spinal cord injury hires at Texas A&M, including mine, and helped us form the Texas A&M Spinal Cord Initiative. So with that, I'll get started on tissue clearing. And I really want to go over kind of the basic principles of tissue clearing and how it works. So as I'm sure most people are aware of, it's very difficult to image through thick tissue, in particular thick central nervous system tissue, because of the opacity that scatters light and prevents light to penetrate deep within the tissue and then prevents light from coming back out of the tissue. And so tissue clearing relies on 2 principles in order to turn a tissue completely transparent. And these 2 different steps are lipid removal, which I'll oftentimes refer to as delipidation, and then refractive index matching. And so if you combine these 2 techniques, you can end up with a fully transparent sample that allows light to pass through with minimal disruption. And so you can now image deep, several millimeters within tissues. And so tissue clearing itself is actually a technique that's been around for over 100 years, but it's recently really expanded and become a prominent scientific technique. And so there are many different techniques that exist today. This is a family tree kind of showing some of the main classes of techniques. You have your organic solvent-based techniques. The disco techniques are really the most common of all of these. You have high refractive index matching techniques. So these techniques actually don't really rely much on lipid removal, but take advantage of refractive index matching and how that can actually go a long ways towards making tissues transparent. You have your hyperhydrating solutions, including cubic, as well as your tissue transformative protocols, the most famous of these being Clarity. And even though this figure is only a year old, this is a Zeiss light sheet microscope that we had at the time. And because it could be performed without any specialized equipment. So you don't need an electrophoretic chamber in order to perform this passive Clarity technique. In addition, it's gentle, which makes it compatible with endogenous fluorescence as well as fluorescent conjugated immunolabeling. And as you can see, it actually does a very good job at clearing central nervous system tissue. So what does that look like? The PAC protocol is a very simple process, surprisingly. You take your tissue and you incubate it with an acrylamide monomer solution that has a thermal initiator. Once that the tissue has been saturated with this monomer solution, you then heat it up to activate the thermal initiator, which cross-links the acrylamide into a hydrogel within your tissue. You then take that tissue hydrogel hybrid and incubate it in SDS solution with a little bit of heat and motion. And over several days, the sample will mostly or if not entirely delipidate, resulting in an optically transparent tissue. You can then equilibrate that tissue in refractive index matching solution to finish the clearing process. And so this can be performed with electrophoresis, but today we're going to be talking about just passive Clarity. So what does that look like in the spinal cord? This is a mouse spinal cord that has been that has the meninges removed and has already had the hydrogel polymerized within it. And as you can see, it's very opaque. It's impossible to image all the way through this tissue. However, with just a few days in SDS solution, the sample becomes almost fully delipidated, and now light can readily pass through. And you can start to imagine imaging completely through this tissue. However, I'm sure some of you have already noticed that there's substantial warping of the light path within this tissue. You can see it really prominently up here in the cervical region. And so this is why you need refractive index matching. By equilibrating the sample in a high refractive index solution, you can make a uniform refractive indices throughout the entire tissue as well as in the surrounding media. And this allows the tissue to become completely optically transparent and light to readily pass through. And yes, there actually is a tissue in here. It's just really clear. So we can then take that sample, and typically we will embed it in agarose and equilibrate it. And we do this inside of a syringe or a capillary. And this allows us to hang the agarose and the sample within our imaging chamber of our Zeiss light sheet. And so this imaging chamber is filled with that same refractive index matching solution. And it has two illumination objectives that bend light into a thin sheet, hence the name light sheet. As you pass your sample through that light sheet, the illumination light will cause the endogenous molecules to fluoresce, and that light is captured by the detection objective. On the right here is a schematic of that. You can see the thin sheet of light that's created by the illumination detection objective. And all of this light is in the focal plane of your detection objective. So all of the emitted light that's collected by your detection objective and the entire field of view is in focus. And so you can actually really just rapidly scan your tissue in the Z direction through that light and collect all of the emitted fluorescence. And because of this, because you don't have to scan in the X or the Y dimension, light sheet is actually incredibly rapid. You can image tissues 100 to up to 1,000 times faster than you could with the confocal. And so I was fortunate early on when I was exploring this process to obtain some samples that were part of a collaboration by my former PhD advisor, Dr. Shelley Sakayama Albert, who was at the time, or who is collaborating with Dr. Stephen Crone. And these were CHX10 Cree mice that had been bred with a Cree-dependent TD tomato reporter mouse, resulting in endogenous fluorescent or endogenous expression of TD tomato in V2A interneurons. I've included a schematic of different interneurons here on the right from a great review by Martin Goulding, showing the V2A interneurons here and the long distance axon extensions that they create in the ventral portion of the cord. And so if you take the cleared sample, which is shown here with the schematic, and you pass it through the light, you can then record the image at specific Z steps as the sample passes through the light, showing here on the left. And as you can see, at each location, the light that's coming in is in focus. And so this is the entire thickness of the spinal cord that has been cleared that we can image all at once. And so that video there is probably sped up about 10 times, but still you can imagine you can record the entire thickness and width in one field of view in about 3 or 4 minutes. You can then take that Z-stack and reconstruct it in three-dimensional analysis software. Here we're using a Mars. And so you can see the axons and the soma from the V2A interneurons. And then create a full three-dimensional video. Right here we're showing one field of view from the thoracic cord from the CHX10 TD tomato mice. And so you can actually, this is actually the first time that we've been able to visualize these V2A interneurons in 3D, and you can really appreciate their abundance within lamina 7 of the spinal cord, as well as these long distance axon tracks that they create running the rostrocaudal axis of the spinal cord. And so this was actually one of the first samples that we had, and so we actually didn't clear the whole cord, we only cleared a portion of it. But we were able to image up and down from about the mid-cervical to the full thoracic dimension of it in order to visualize these long-distance axon tracks that could be coming from V2A interneurons within the spinal cord, as well as could be descending axon tracks coming from the V2A interneurons in the brainstem, as Jared had described a few weeks ago. And even with these preliminary samples, a talented research assistant in Todd McDevitt's lab, Esther Mihaly, was able to develop an analysis pipeline in which she could quantify the number of V2A interneurons using their soma at different regions within the spinal cord. And all this image processing and quantification was done in Amaris. And so we're really fortunate to continue this collaboration with Dr. Stephen Crone, and actually received a bunch of new samples that we were hoping to image the entire spinal cord as well as the hindbrain. But we got these samples the week before the shutdown, so they're sitting in our fridge, and we're really excited to get back in the lab and analyze these. One of the things that we have been doing during this time remotely is working on computational analysis methods, including fluorescent-based tractography analyses that can help us analyze these long-distance tracks within these images. So in Todd's lab, simultaneously with these experiments, one of his grad students, Jessica Butts, who also happens to be my better half, had worked out a protocol for differentiating V2A interneurons from human pluripotent stem cells. And as part of this publication that came out in PNS a few years ago, we had transplanted about 500,000 of these cells over 4 sites in the thoracic level of the mouse spinal cord. And we left these cells, or we left the cells in there for 14 days before isolating the spinal cord and performing sagittal sections. And so this is a figure from that paper showing the transplant site. And the cells are identified with 2 markers, STEM121, which is a cytoplasmic marker, and human nuclear antigen. And what we could see are these long distance processes as much as 5 millimeters down the spinal cord that had these thin-like projections that we assumed were axons. However, we could really only see these extensions for short distances because they appeared to be weaving in and out of the section. And so we were really interested in using tissue clearing and lightsheet imaging to better analyze these transplanted cells. And when we first started out with this, we used STEM121, but we quickly realized that it was not compatible with the clear tissue. We couldn't get any positive staining with it. And we were fortunate to find a separate marker. Oops, get the video playing here for you. It doesn't seem to be playing, but we'll move on. We were able to find a separate marker, human tau. And so this was an antibody against a human-specific version of tau. And as you can see, it labels the cells within the 4 transplantation sites very well. And so with this, we can start to analyze axon extension up and down the spinal cord, and looking at the entire transplant architecture without the need for tissue sections. And so I'll move on because we have another video that shows more of that. But with these results, we were highly motivated to see what this looked like in the injured spinal cord. So we performed a moderate contusion injury in skid mice. We waited 14 days and again transplanted 500,000 cells at the lateral edges of the lesion site. We waited 14 days and performed sagittal sectioning just to confirm that the cells had survived within the injured cord. And we found many STEM 121 positive cells. We could see these thin axon-like projections that were extending around the lesion site, typically through spared tissue. We could see many STEM 121 positive cells that also co-expressed NUN, so confirming that we did have neurons within our transplanted population. And then we could even see processes or cells extending along or through the glial scar around the lesion site. However, because of the thin nature of these tissue sections, we really could only follow them for short distances. And so we were really interested in seeing how this worked with tissue clearing. And so this is a video of a single field of view from a 5x objective showing the H-tau labeling on these transplanted human neurons. This section or this tissue is counterstained with NUN, so you can see the lesion site where you have the absence of your NUN positive cells. And so you can really start to visualize the axon projection that's coming from the transplant sites where we had the greatest survival. And so these are able in as little as 14 days to completely extend around the SCI lesion site. And this is something that would be fairly hard to pick up just looking section by section, but is readily apparent with our 3D videos. So at this point, I'll take a brief pause and answer any questions.
Yes, and we have quite a few questions coming through. A few are getting fielded in the chat, so if you want to take a look at that, feel free to scroll on up and down. But a couple that haven't been addressed yet. One is from Julian Bouvier, and he asks, what index matching solution do you use?
So we make our refractive index matching solution, or RIMS, from nicodens, which is just another brand name for histidens. And so they're both the same chemical. We just found a cheaper source for nicodens, and so that's our main refractive index solution, which we measure to a refractive index of about 1.48.
Okay, great. We have another one from Dinah Howland. What's the maximum rostral to caudal length of spinal cord specimen that can be imaged?
That's really dependent on the microscope more than anything. For us, with the current configuration with the Zeiss chamber, it's been about 2/3 of mouse spinal cord, but Zeiss just released an update in which they have a deeper chamber. So we're now hoping to image the entire mouse spinal cord from top to bottom.
Okay, one from Timothy Fall asks, the delipidation process removes myelin, correct?
It does, yes. Or to be more correct, the lipid component of myelin.
Okay, I'm trying to keep up with all of it here. We have some questions coming in from the Petruska lab. Can the entire length of the cord be imaged with light sheet projection and cross-section by lowering the cord through the light sheet?
So you can visualize the cross-section from the three-dimensional image. And if you perform multi-view imaging, you can get the cross-section in high resolution. So typically with this, your XY is your highest resolution and your Z plane is the lowest resolution. And so it really, it depends on the setup. So you could image from the dorsal or the ventral view and then use the 3D software in order to look at the cross sections. If you wanted to take a thick several millimeter chunk of spinal cord and lower it down in there, you could image directly through the cross-sectional field. So there's kind of a lot of flexibility in how you do it.
Okay, we just had one come through from Murray Blackmore. How long does fluorescence last with PACT?
So I'll ask a follow-up question on that, and whether, Murray, if you're referring to endogenous fluorescence or fluorescence that's conjugated to antibodies. As far as the endogenous fluorescence, once it's in the rims, it actually, you can store it in the fridge and it lasts for a long time, weeks to months. We really haven't tested the decay of the endogenous fluorescence. And then how you perform the delipidation steps will greatly affect it. So we try to keep the temperature at 37. You could go up to 60 or to 80 degrees Celsius if you wanted, but you're going to lose, it's going to start to damage that endogenous fluorescence. There are also different fluorophores that are more stable, and I'll show some examples of some that we've been using that work really well with the clearing process.
All right, great. Keep the questions coming in, and we'll have another session at the end.
Okay, so when I started my lab at Texas A&M, I was really interested in other neuronal cell types that we could image within the spinal cord, in sort of this effort towards to move towards a map of the spinal cord. And so one populations that we were really interested in were the cholinergic populations, including the motor neurons as well as the V0C interneurons. And one of the antibodies that works really well in this process is choline acetyltransferase. So here I'm showing a video of the Z-stack of the light sheet image through it, in which you can see several distinct cholinergic populations within the spinal cord, including the motor neurons in the ventral horn here, as well as their axons heading out to the periphery. And then you can take that Z-stack, and then this is the three-dimensional video from that Z-stack. We've also counterstained with Nu-N, shown here in green, to kind of help orient the chat staining within the spinal cord. And so from this image, you can clearly see the motor neurons as well as their axons. They're heading out to the periphery. You can see different cholinergic populations here at the midline, including the V0Cs, as well as potentially motor neurons that might be part of the intermediolateral nucleus. And then interestingly, we found this sparse dorsal cholinergic interneuron population that we really hadn't considered before, but we went in the literature and we actually found several publications that have talked about this population, both within rodents as well as in non-human primates. And so then we were really excited. We were able to get, it didn't require a transgenic mouse, but we were able to label these distinct populations. However, one of the limitations that we came across is this chat staining really only labels the cell bodies and their axons. And if we wanted to do a more detailed morphological analysis, we needed to have some fluorescent reporters that could fill the entire neuron. And so we've actually played around with a lot of different ones, including rainbow and confetti mice. And what we realized is we actually needed to kind of create our own reporter for our specific, for the task that we were hoping to accomplish. And so we're fortunate that we're lab neighbors with Dr. Jen Doolin. And so she has worked a lot with AAVs and has been part of a project that developed a new set of fluorescent proteins that were specifically designed to withstand clarity-based procedures. And so we used the new serotype of AAV that can cross the blood-brain barrier, and we use that to deliver these common fluorescent proteins. We did this by retroorbital injection of this vector into chat-free mice. We then waited 4 weeks and performed tissue clearing. So you can see the chat immunostaining here for that mouse. And then for some reason, it's not playing the video. I'm going to exit really quickly. Oh, there we go. So this is showing the immunostaining, and then soon we'll turn on the red channel, and you'll be able to see how this comet vector recapitulates that chat staining, because it's a pre-dependent vector that's injected into chat-free mice. But it also does a very good job of highlighting the detailed morphology of these cells. And so we'll zoom in here. And so this is just with a 5x objective, but you can already see the fine detailed morphology of these cells that are labeled with this common vector. And so unfortunately, with the shutdown, we've had a bit of a pause on this work, but we're really interested in optimizing this vector and creating new variants that can help us accomplish a variety of tasks and looking at different populations within the spinal cord. So much of my postdoc work and much of my current lab focuses on acute inflammation following spinal cord injury. And we've been particularly studying neutrophils, which are the most abundant immune cell type within human blood. They're the first circulating immune cell type to invade the spinal cord in large numbers. And it's already been worked out that neutrophils peak near the end of the first day following spinal cord injury. However, this data was generated predominantly with flow cytometry in tissue sections, which result in either complete loss of spatial information or incomplete spatial information because you're taking every 10th or every 20th tissue section. And so we're really interested to see if lightsheet could help us better understand the distribution of neutrophils within the acutely injured spinal cord. Now, with every new technique, there's always challenges. And for us, we found that our main surface marker for neutrophils, Ly6G, was not compatible with cleared tissues. We were able to identify an antibody against MRP8, which is a cytoplasmic and granular protein in neutrophils, and use that to label neutrophils within the injured spinal cord. We have neutrophils that seem to be present in the meninges that we can digitally isolate from those that have accumulated in the spinal cord using a MARIS. And then we can use a MARIS to quantify the number of neutrophils within the spinal cord and look at those numbers as a function of the length and width of the spinal cord lesion site. And what we found is that these MRP8 positive neutrophils, so these are neutrophils that haven't degranulated, are accumulating at the lateral edges of the spinal cord injury site. Here's the details on the injury over here. And so this is really exciting because it was kind of giving us a new view into the behavior of neutrophils within the injured spinal cord. And we could take those images and perform maximum intensity projections to look at all of the neutrophils and neurons within the spinal cord. And here you can see, this is kind of a bird's eye view looking from the dorsal side of the spinal cord. You can see the lesion site where you have loss of your new end, as well as your MRP8 neutrophils accumulating at the edge of the spinal cord injury. As you move from the lateral edge towards the center of the SCI lesion, you can see that there seems to be a decrease in MRP8 intensity within these neutrophils. So this might be suggesting that we have degranulation of these neutrophils within the SCI lesion. We've been really fortunate that we actually have a neutrophil reporter mouse in our lab, and we had just started these experiments before it shut down, and so we're really excited to get back in and be able to look at all of the neutrophils within the injured spinal cord, as well as adding in this MRP8 stain to look at degranulated versus non-degranulated neutrophils. You can even take single Z-planes within here and look at these non-degranulated neutrophils, and we found many instances near the injury site where these neutrophils appear to be interacting with the soma of neurons. Now, we don't know if these are causing damage or if they are reacting to already damaged neutrophil or neurons, but this gives us a starting point to look at these interactions. And then just to confirm our results, we actually performed flow cytometry on neutrophils and looked at the presence of MRP8 and another granular protein, neutrophil elastase. And we found that actually many of the neutrophils that we can isolate from the injured spinal cord at 24 hours post-injury are not degranulated. And so there are definitely degranulated neutrophils in there, which we believe would be in the lesion cavity, but it was actually a kind of a new finding for us to realize that many of the neutrophils within the cord are not degranulated yet at the peak accumulation time of 24 hours post-injury. Okay, so I'm going to finish up really quickly just with some last work that we submitted recently. This was work that was done in collaboration with the late Wes Thompson. It was done with a really talented graduate student in his lab, Ryan. And Ryan was really interested in quantifying neuromuscular junctions and mouse models of muscular dystrophy. However, the bungarotoxin staining that we use to identify neuromuscular junctions is not compatible with PAC clearing. He was able to show if you just do refractive index matching of muscles, you can actually make them fairly transparent, and that is compatible with bungarotoxin staining. So you can see here that he can actually label all of the synapses through a muscle by just doing refractive index matching. And then he was able to use that technique to look at the degeneration of neuromuscular junctions in these muscular dystrophy models, mouse models. In addition, we could take these muscles following PAC clearing and perform chat staining similar to what we did in the spinal cord. And so you can start to see the distribution of innervation of these motor axons within the muscle. So quickly wrap up, I want to say thanks to everybody again. Special thanks to Todd McDevitt, who let me explore these projects within his lab, the members of my lab, our funding sources, and especially our collaborators and the imaging cores that have helped us out. And I have my email down here if you have any questions that you would like to follow up with. And at this point, I'll take any remaining questions.
Okay, thank you, Dylan. So an outstanding question from the Petruska lab was, is the spatial resolution sufficient to detect axons in cross sections, which could then be reconstructed to see axons transverse to the length of the cord?
Yeah, so most of the research that I've shown here is just with the 5x objective, because you can capture the entire width of the spinal cord in one field of view. The system also has a 20x objective if you wanted to get higher magnification, but you would have to stitch across the entire width of the spinal cord, but it's perfectly possible with these clear tissues. With that 5x though, it's a high numerical aperture objective, and so we were able to get axonal resolution within our, the one video wasn't playing that actually shows it best. As far as in the Z dimension, it would be best if you did multi-view acquisition to basically get the XY resolution throughout the entire image. And that would provide you with sufficient resolution to look at axons.
Okay, another question from Lynette Montgomery asks if antibodies for IHC actually penetrate further into cleared tissue.
Typically, it takes a lot longer for antibodies to get into cleared tissues. There's a lot of things that you can do to boost the diffusion rate. But typically, when you're staining these cleared tissues, you're looking at staining times of 2 to 3 days per round of antibody. And you also need a lot more antibody because of the greater amount of antigen that's present there. Certain antibodies such as GFAP, you could put 5 vials of antibody on and not stain all of the antigen. There are new methods, so I pointed to eFlash, which is a new method out of the Chou lab at MIC that has shown how you can sub-maximally label tissues. And so that's something, if you're interested in antibody staining in clear tissues, I highly recommend starting with that paper.
Okay, next, Sabia Rana asks, if we don't have access to lightsheet microscope, can we rig chambers to image packed cleared tissue on confocal scopes? Absolutely, yeah. And so it's this is typically the method that's done for organic-based clearing. There are specific lightsheet microscopes that work really well with the organic solvents found in distal clearing, but in many cases they get kind of embedded in modified slides in order to prevent the lenses from actually touching the organic solvents. And you could do a similar thing with these pack cleared cords, though the rims, the refractive index media is not damaging, and many of our 20x objectives are dipping objectives. Okay, so a question started with Timothy Fall, but a whole conversation has blossomed about membrane tagged fluorescence or other ways to actually image myelin. Yeah, so while we're on that, let's just move on to the part of the slide that I didn't have time to get to. So there was a paper that was published out of the Chou lab a few years ago that introduced this staining called switch. I would say for antibody staining, it's been updated in the eFlash manuscript, so I recommend going there for using this for antibody staining. But for myelin staining, what they showed is if you add a little bit of SDS in with a lipid dye, such as dye D, you could get uniform labeling of myelin within the brain. And so we've actually been able to accomplish that within the spinal cord. So even though PACT is a delipidation process, there's enough residual lipid that you can actually label the myelin with these lipophilic dyes. And if you use the switch technique, you can get uniform labeling. So we've done it both within the uninjured cord, but as well within the injured cord. Let me get this started.
And so we're really hoping to apply this technique to start looking at white matter damage following spinal cord injury. I will say, though, that we don't think that this is capturing all of the spared white matter, because there is substantial removal of the lipids with the clearing process. So now we're looking for specific myelin-associated proteins that we can perform immunolabeling with to get a better idea of what the white matter damage looks like after spinal cord injury.
You're getting a lot of compliments on these slides particularly, and the rest of your slides. So great work all around. Another question, Lawrence Moon would like to know what proportion of neutrophils at 24 hours are not degranulated, and if you expect that they'll all degranulate if they're within the parenchyma rather than blood vessels.
So our quantification suggests that about 70 to 80% of the neutrophils at 24 hours are not degranulated. That is the question that we're trying to figure out, is whether these are on their way to degranulating, or whether it's possible for neutrophils to actually egress out of the spinal cord and leave without undergoing degranulation. And so that's something that my lab is interested in looking at, and how that proportion changes as a factor of time following injury. But we were actually shocked to find that many neutrophils that weren't degranulated, even though our flow cytometry shows that all of these neutrophils are activated. They've all shed their L-selectin, they all have higher levels of CD11B, but they're not degranulating. And so that may be that they never go down that particular pathway, and that they actually leave the spinal cord and go elsewhere.
Fantastic. Thank you, Dylan. And also thank you to Abel and Austin for our earlier presentations. We have plenty of questions that have come up in the chat, and I think that Dylan's going to be sticking around in the chat a little bit after this if you have further questions. But thank you very much. So next, I'd like to introduce Tuesday's seminar series. Is going to be Dr. Andrew Gaudet, which you might have seen in the chat earlier. He is from the University of Texas, and he's going to be talking to us about spinal cord injury disrupts the circadian metabolic axis and salience of pain versus anxiety. So tune in next week at 2:00 p.m. Eastern Time on Tuesday.